Thank you, Andrea. Can you hear me okay?
Yes.
Just at the beginning, I would like to thank Nanyan for organizing this webinar and for inviting SB to give a talk today. So as George and Andrea said, I'm going to be talking about utilizing SURFE2R for TMM 175 studies here at SB. Sorry, I think my laptop was lagging. So I think it's needless to introduce TMM 175. It is an exciting new target with therapeutic potential. And here at SB, our aim is to develop robust reagents and screening cascades to facilitate TMM 175 drug discovery screening. And for that, we have a few cell lines prepared with a wild type TMM 175, human one, as a cell line, as well as two variants, loss of function and gain of function variant, and on the way, mouse and rat orthologs of TMM. But before I hop on the SURFE2R train, I would like to briefly talk you through our journey with TMM 175 using electrophysiology platforms. So our aim is to develop a robust cesium-based automated electrophysiology assay, and we achieved that with wild-type TMM 175 being expressed in inducible hex cell line. And as you can see, the TMM 175 in inducible cell line, we pretty effectively created a baseline, which then could be inhibited by the 4AP in a concentration-dependent manner, and then it could be activated by zinc chloride, again to be blocked by 4AP. This was validated in the cell line with not overexpressed TMM 175, and we can see any appreciable current apart on the untransfected cell line, which was good. And using automated patch clamp, we identified novel TMM 175 modulators. So we screened about 20,000 compounds, and using this assay, we were capable of identifying both activators and inhibitors. And fluorescence-based assay showed a great correlation with our APC assay, and thanks to it, we successfully measured activity of hit compounds and reference compounds with those two platforms as cross-referencing each other. So here's just a table with chosen hit compounds, TMM activators in our fluorescence and electrophysiology and their EC values for them. But of course, we know, Andrea mentioned that as well, that TMM 175 is not just a potassium, cesium, or rubidium channel, it's a proton leak channel in lysosomes. It's highly proton selective, and it's gated by luminal protons, and mediates lysosomal proton efflux. So here at SB, we wanted to develop this assay to measure the protons using automated patch clamp, and we developed an electrophysiology assay to measure inward proton conductance. And we found this sweet spot, as we call or refer to, at pH 6. So this just represents a pH CRC of TMM 175 and an application of different pH values going from 7.2 to all the way down to 4.5. And hopefully you can see that the lower the pH, the higher the proton conductance. And now what I mean by the sweet spot at pH 6. Well, we know that TMM is activated by the pH itself, and it's also activated by the DCB-IB compound. So we kind of chose this point based on this data, where activation by pH doesn't quite reach the maximum capacity of TMM itself, and where it's still at this same pH can be activated to its maximum by the DCP-IB. So based on this, we developed an assay, a low pH assay, where we measure proton conductance stably transitioning from pH 7.2 all the way down to pH 6. And hopefully you can see that as soon as you drop the pH, the amplitude recorded is larger, and it's a pretty stable current, and it is then can be blocked by 4AP. And this block by 4AP is pretty consistent and is concentration dependent as well as visualized on this graph here, but also with EC50 value over here. And a good thing was that no effect on untransfected cells was recorded during those conditions. So we took this assay a little bit further, and as opposed to blocking the signal, we wanted to enhance the proton conductance, and we did that with the use of DCPIB as well as our preferred SB compound. And we found that EC50 values were pretty consistent with the original screening assay. And here's just to represent that DCPIB, it was pretty consistent and concentration dependent when pH 6 was dropped and different concentrations of DCPIB were applied. It was nicely plotted on the graph with a nice EC50 value, and all those currents were blocked by 4AP. Similar situation with our SB compound, also quite a nice concentration response curve. And yeah. But of course, all this work was done on live cells. And while lysosomal patch clamp is possible and we will be working towards it as well, it's quite challenging at the moment and not really cost effective. So other electrophysiology platforms needed to be explored. And that's where solid supported membrane electrophysiology, SSME, comes into play. And of course, I'm not going to be going into any details because we had George and then Andre explaining it very well, all the technology, but let me just briefly recap the advantages of using SSME for those purposes. So no living cells are required. As Andrea mentioned, those are just membrane stored at -80, so no continuous culture needs to be performed for those assays. SSME measured transport of charged ions, and it's ideal for studying electrogenic transporters. Its high stability of the sample allows multiple repeat experiments, as Andrea said, on SURFE2R, up to 20 measurements are reproducible and there's no loss in currents in those circumstances. And of course, it's suitable for studying lysosomes, which what we're interested in. So apologies in advance for this quite busy slide, but that's just to recap what Andre was saying, and I'm showing you this just to prove a point of sample validation. Sample of TMM175 that we prepared here at SB using our own cell line and validating it on SURFE2R here in-house as well. So just very briefly about selectivity of TMM175, not to such a great detail as Andre did, but we do see similar behavior where cesium is a preferred ion here in green over potassium in orange. And also this is just a 50 millimolar of potassium on the graph in this case of the untransfected cell line that we used for preparation of our lysosomes as a negative control. And you can see there's no current recorded or a negative current recorded at those conditions. EC50 values for both potassium and cesium were pretty consistent as well, meaning that the assay was working very well. And then again, just as principle, we tested known blockers and activators on our sample made here in SB, and we did a concentration response curve for AP, and this is just a graph representing what it looks like on SURFE2R as well. And we also performed some zinc block as well, which was also pretty successful. Then, of course, we also wanted to test some activators, and the preferred one and known for TMM175 is DCPIB. And you can see a nice concentration response curve. Again, just a graph representing what it looks like on SURFE2R on the currents. And we also chose another activator, ML6733, which is a known activator for K2P potassium channels. And while the CRC is looking pretty convincing, it is in a 2-digit micromolar range here, about 15 microliters, but it doesn't fall far from the EC50 values for K2P transporters as well, which range between 20 to 30 micromolar EC50 value. So once we had our sample validated on SURFE2R, our own sample made from our cell line, and we were pretty happy with the results, and they were comparable to what Nanion was seeing as well, we could take our screen, compound screen on SURFE2R further. So we found a few candidate compounds activated TMM175 on purified lysosomes, with an example here on the right. But strikingly, some of the compounds were not activating TMM175, including our preferred SB compound that we've been using routinely in electrophysiology, as well as on fluorescence assays. So kind of to understand why this could be the case, why we've seen so discrepancies between the compounds and testing, then between on electrophysiology platforms and fluorescence, we did some additional tests. And for that, we tested another compound, known compound to be activated TMM175, which is SC79, along with our SB activator. So SC79 is known to be working or assumed to be working on TMM through AKT pathway, so not directly. So we kind of thought this could also be the case with our SB activator and other compounds that are not activating TMM on SURFE2R. So this is just a graph with average peak currents representing different concentration of both activators, and you can hopefully see there's not much happening, not much activation apart from the background noise is going on. And as I said, SC79 works on TMM through AKT pathways. So we did a fluorescence study where we used thallium as a proxy to measure potassium translocation on live cells. So this is just no compound added, just fluorescence based on only on cells. Then we added our SB compound, and hopefully you can see the activation happening. Same happened with SC79, nice activation comparison to no compound bar. And again, we tested DCP-AB as a kind of positive control too, also seeing a good activation. So SC79 obviously activated TMM175, and then we wanted to see if it really can be reversed by adding AKT inhibitor. And yes, that was indeed the case. You can see that with addition of inhibitor, the fluorescence dropped almost to reflect the no compound fluorescence. That, however, was not the case with our SB compound or even DCP-IB. What kind of made us believe and assume that it could still be working on TMM, but through a different pathway, still indirectly, but through a different pathway. And that kind of presents SURFE2R as a great cross-reference tool between different platforms. So something that other platforms are not, cannot provide at the moment. And having this in mind, that provides and opens up new and exciting avenues for the drug discovery, where we can at least to some extent say, well, those compounds, not just for TMM175, but other transporters as well, whether the compound works directly on the transporter or it's working through another pathway. So then, of course, we know that TMM175 is influenced by the pH, and here's just to reflect 50 millimolar potassium flux at different pH values. And hopefully you can see that with lowering the pH, the potassium translocation drops. So that's something that, of course, Andrew went into more detail, but that's something what we also want to explore here at SB. So having this in mind, we also wanted to see what native environment of the liposome would look like on SURFE2R. So for that, we internally acidified the inside of the liposome and applied currents, applied a swift change of external pH. And with that, we observed that internal acidification potentiates the permeability of TMM175 to potassium. So here we have the average peak currents adjusted with untransfected currents for the pH 7.2 at 10 millimolar potassium. And nice reproducible currents. And then once we switched to the pH gradient measurements, meaning that we acidified the inside of the liposome to pH 6, and then swiftly applied different pH of 7.2 in this case with 10 millimolar potassium, there was an increase in current, which was pretty consistent. And the percentage increase was roughly about 15%, give or take, pretty consistently across different sensors. Then we wanted to take this even further and see if we can potentially DCPIB. So what we did, we repeated the same experiment, but now with the addition of DCPIB with the EC value obtained with CRC. So hopefully you can see that the gradient measurement without DCPIB is potentiated by DCPIB, and the percentage increase were roughly about 50%, again, quite consistent across different sensors measured on different days as well. And using similar setup, we can also track the proton currents on SURFE2R for TMM175. Again, proton currents were measured by the application of swift exchange of external pH. In this case, in none of the solutions, potassium was present, so it was just application of non-activated solution to adjust the lysosomal pH to wanted pH, in this case, either 6 or 4, and then application of, again, non-activated solution at a pH 7.2. And a larger conductivity was observed when using acidic solution internally. So here we have the H conductance using non-symmetrical pH gradient, pH 6 inside, 7.2 applied externally, in comparison to pH 4.5 and 7.2, you can see a massive increase in the proton conductance. Again, as Andre explained, those currents are adjusted to what we are seeing on untransfected, so this reflects only the translocation of protons via TMM alone. So now kind of the last thing I want to tell you about is the preliminary data that we got for a loss of function mutant variant of TMM175. And our initial data shows that variant displays concentration dependent activation by potassium with EC50 comparable to the wild type. And TMM loss of function also shows a clear activation by DCPIB. And here's just a couple of concentrations tested at pH 7.2 with 10 millimolar potassium present. And those conditions in this current can be blocked by 4AP as well, as shown in green here. So now just to kind of wrap everything up and summarize, I want to share quite an exciting news as well. The acquisition of SURFE2R 96 platform for a high throughput compound screening, not just TMM, but other transporters, and the fact that we're expanding protein transporter research as well. So here's just the list of neurotransmitter transporters that we have on the shelf right now that are suitable for the SURFE2R analysis, but we have another 20 plus targets on the way that are being under development right now, which is quite an exciting news. So again, just to kind of summarize and wrap everything up, our TMM175 research, we have well-established fluorescence high throughput screening techniques, as well as automated electrophysiology assays for proton and potassium conductance as well. And now we also have SURFE2R, which is quite a powerful tool for cross-reference between those two platforms. We validated the assay for potassium and proton conduction using SURFE2R, and we also now tested loss of function variant, and we have an assay ready for your research needs. We are also acquiring a big SURFE2R 96 one for a large screen compound, with which we will be expanding our protein transporter research too. So again, I would like to thank Nanyan for organizing this webinar and for inviting SB Drug Discovery to give a talk. It was a pleasure for me to represent SB Drug Discovery. Of course, we will have a Q&A session just in a minute, but if you forget about anything and you want to get in touch, you can send me an email directly or find an inquiry email on our website. So get in touch, and thank you very much for your attention. Thank you for having me.